Chapter 8, Microbial Genetics. So this cover slide is showing one of the processes that we will learn towards the end of this chapter, and also that we will carry out a lab exercise on. It's called transformation. And transformation is the process by which bacteria can take up DNA from the environment, and if they incorporate that DNA and do gene expression of that DNA, if there's a new gene on there that they didn't possess before, then the bacterium gets a new trait. So in this example, the bacterium has picked up this DNA and now it has produced fimbriae. But before we get to what's called horizontal gene transfer, which includes transformation, we need to cover the basics of genetics. Just a little introduction about plasmids. We've seen them here and there, I think, so far. So remember that bacteria have a single, meaning one, double-stranded circular chromosome for their DNA, but they also can have additional little pieces of DNA that are also circular, double-stranded, and but they're only about 1 to 5% the size of the bacterial chromosome, so much smaller than the bacterial chromosome, and they can carry extra genes for antibiotic resistance, genes for like extra metabolic capabilities, genes that encode virulence factors, which make the bacteria more disease-causing. So this is an electron micrograph of some plasmids. So you can see these are little pieces of DNA here. So again, we're kind of going big picture, and then we will start honing in on some details. But it's very important to understand this central dogma of genetics, or really of life, because this is all living organisms follow these rules. So DNA is the genetic information of living organisms, and that DNA has to be transcribed into RNA, and the RNA gets translated into protein, and the protein is what actually gives the organism its characteristics, its functions, etc. And in this chapter, we'll also talk about mutation and how mutations are defined as a change in the DNA. But if the DNA is changed, that means the RNA that's made from it will be changed. That means the protein that's made from it will be changed. And that means that there could be altered function of that organism. So we will go through mutations later, so you don't really have to worry about this diagram right now, but it's just kind of giving you an overview of some of the types of mutations that we will be talking about. And again, just still kind of looking at the big picture. Why is this chapter important? Why is it important to understand genetics? Well, because it plays a big role in virulence and pathogenicity of microbes. It plays a big role right now in issues with antibiotic resistance. It's important for us so that we can control disease, so that we can create medications as well as vaccines. So you can see some examples here of how genes and gene expression impact our lives with diseases, biofilms, antibiotic resistance, biotechnology, which is the production of medications like insulin, as well as vaccines. All right, enough of the big picture. Let's start getting into this chapter. So just starting out with some terminology. So genetics, right, that's what this chapter is called, is microbial genetics. So it's the study of genes, how they carry information, how the information is expressed, and how the genes are replicated. Chromosomes, that's the structures that contain the DNA. It's kind of how is that DNA organized. So bacteria, prokaryotes, have one chromosome, right? We said they have one single I hate using the word single because I think you want to think single-stranded. So one, but it's double-stranded circular chromosome versus other organisms like ourselves that have linear chromosomes, right? Ours are not circular, and we definitely have more than one. I wonder how many of you know how many we have. I think a lot, because you've gotten this information before, right? So we have 23 pairs or 46 total chromosomes. Are we the organism with the most chromosomes out there? Nope. There's plants, there's insects, there's all kinds of things that have more chromosomes than us. So chromosome number does not equal complexity. It just depends how the DNA is organized. You have different chromosome numbers for different organisms. Genes, so same thing, kind of depends on how that DNA is organized into these genes. Different organisms have different numbers of genes. It does not necessarily reflect complexity, but I mean, it does to, I guess, a certain extent, usually. You know, the lesser genes an organism has, the simpler they are, the more genes they have. They tend to be a little more complex overall. That one you can kind of generalize. And genes are just segments of DNA that code for functional products, which are usually proteins, but we do have some RNA that's encoded by DNA that does not make proteins. Anyone think of any RNA that does not code for proteins? So if you remember, there's 3 types of RNA. You have mRNA, rRNA, and tRNA. So mRNA is the RNA that is translated into proteins, but rRNA and tRNA are not translated into proteins, but they are important and they do work in the process of making proteins. But those would fall into a functional product that's, encoded by DNA, but it doesn't actually make a protein itself. Genome is all the genetic information in a cell. Genotype is just kind of referring to the whole genetic makeup of an organism, kind of what genes do they possess, and phenotype is the expression of those genes. So again, central dogma, don't forget this. It's very important. This is what a big part of this chapter will be going over. We'll go in detail through the processes that these arrows represent. Transcription and translation, right? We'll be going over that quite a bit. So I really like this picture. It is a picture of E. coli bacterium here, this little rod-shaped bacterium, which it looks quite big here because it's under an electron microscope. And what they've done is they've disrupted the cell, disrupted the DNA. The DNA has become unraveled and spilled out. And so it's crazy to think that this is just one double-stranded circular piece of DNA that makes up the bacterial chromosome. And how in the world does all of that fit inside of that bacterial cell? Well, it's usually highly compacted and supercoiled inside of the bacteria. But again, for this picture, it has been disrupted and spilled out. If you were to stretch out all of the DNA into linear format out of E. coli, it would be 1 millimeter long. And we could see 1 millimeter with our eyes. It's like the size of like a flea. And but you can't see E. coli with your naked eye, right? It's like 1/100th of a millimeter. So how in the world can all that DNA fit in there? Well, again, it's very thin, it's compacted, it's super coiled. If you took all of the DNA just in one of our cells, if you took all of our chromosomes and you lined them up linearly, it would equal 6 feet. And we have, I forget now how many, is it trillion something cells in our body, whatever it is. It's a huge, huge amount of cells in our body, right? And just in one of our cells, you spread out that DNA, it equals 6 feet long. Most of us are not even 6 feet tall. So again, just showing that the DNA is very thin and it's highly compacted and super coiled to fit inside of the cells. So this is kind of a big picture overview of what this chapter is going to focus on. And I'll tell you kind of in the order that we're going to focus on them because this diagram doesn't necessarily go in that order. First, we're going to be covering replication. So DNA replication, it's probably something you've learned before. Why? Why do we have to cover it? Well, because if a bacterial cell wants to divide, which is what they tend to do, right? And when they're causing an infection, they're growing and dividing in your body. The first thing they have to do is replicate their DNA so that their offspring receive that genetic information as well. So it's important to understand DNA replication, not even just for bacteria, but in general for our health, for viruses, and any organism that has DNA, they replicate their DNA, and that is a way that we can focus on treatment of diseases, is stopping the organism from replicating their DNA. So we will cover replication. Then we'll cover gene expression. And again, this is very important because it is that central dogma. All living cells undergo gene expression, and that is where the DNA is expressed into protein through the processes of transcription and translation. So we'll focus a good amount of the chapter on gene expression. And then lastly, towards the end of the chapter, we will cover recombination, which we started out, I mentioned on the cover slide. Examples of that, it's called horizontal gene transfer, and one example is transformation, but there's also transduction and conjugation that we will cover when we talk about recombination. But starting out, we're going to first start with replication. Before we get there, we have to make sure we understand the structure of a DNA molecule. So DNA forms a double helix. So generally you always find it double-stranded and in a helical format. So like a spiral staircase, right? And it has a backbone which consists of deoxyribose phosphate. What is that? It seems very complicated. Well, ribose is a sugar, and it's a 5-carbon sugar. Deoxy just refers to, lack of an oxygen, right? Compared to regular ribose. And phosphate, it's just phosphate group, which is a phosphorus with oxygens on it. So it's not that complicated. It is chemistry. That's why chemistry is a prereq for this class, right? So that's the backbone of the DNA. We'll look at a diagram of it in just a minute here. And then, so you do have those 2 strands. So there's 2 backbones. And on the backbones, you have the nucleotides that are kind of holding those 2 strands together, because you have 4 nucleotides. You have A, adenine, T, thymine, C, cytosine, G, guanine. And A's always pair with T's, or vice versa. T's always with A's, and C's and G's are always paired together because they pair via hydrogen bonds. And so that's what holds the 2 strands of DNA together, is that base pairing. And the strands are anti-parallel. We have to look at a diagram to really understand what that means. And it is the order of these nitrogenous bases or nucleotides that determine the genetic information of the organism. So, A, T, C, C, G, A, T, G, C, we could go on and on and on, right? Just with those 4 letters, that's what makes genes. And genes make proteins, and proteins make function and characteristics and physical traits and, all that. So it's crazy to think that it's really just the sequence of those 4 nucleotides that determines everything about an organism. So here's the diagram. So here is the backbone. See, on this side and this side, right? Because again, it's a double-stranded molecule. So this is the deoxyribose phosphate. Deoxyribose phosphate. Deoxyribose phosphate. Same on this side. But see, it's upside down. It's in the opposite orientation. That's what the anti-parallel refers to. So on carbon-based molecules, you might remember this from chemistry, we number the carbons. And so this is carbon 1. 1, 2, 3. And so the numbered carbons, right? 3, 4, 5. It's a 5-carbon sugar. So we number the carbons. And so that's what this 3' end OH hydroxyl group, right? Over here, the 5' end is the one that has the phosphate. And notice how it's 3' end here, but 5' here, and vice versa up here. So that's that anti-parallel nature. We'll talk a little bit more about it in a minute, why it's important. But look in the middle now, here's your nitrogenous bases, your nucleotides. A's are paired with T's with 2 hydrogen bonds holding them together. C's are paired with G's with 3 hydrogen bonds holding them together. So here's another view of a DNA molecule, more showing you that, a little bit of that helical nature to it, like a spiral staircase. And we're also here going to start looking at DNA replication. So the way DNA replication works is that one strand serves as a template for the production of a second strand. So what happens when an organism is going to, go through reproduction and divide themselves? So the first thing they have to do is copy their DNA. Well, the DNA is highly coiled and, compacted. So first thing that has to happen is there's a bunch of enzymes that have to come in and help relax the DNA. So there's a couple here, topoisomerase and gyrase, that are relaxing the strands here to allow the DNA to kind of get opened up a little bit to allow the replication process to occur. And then helicase, this enzyme actually comes in and actually opens up the DNA, like pulls the strands apart. These two purple, right, are the original parental strands where you see up here, where they're still base paired together with their C's and G's and their A's and T's. But here at the replication fork is where the two strands have been pulled apart. Like think of unzipping a zipper, right? That's been unzipped. And that forms the replication fork. And so then a bunch of other enzymes that are involved in DNA replication, which we'll see mentioned in a minute here, they come in and they start reading the DNA sequence on the parental strand. And it's just based on complementary base pairing. So wherever there's a T, an A comes in. Wherever there's a G, a C is opposite. A, T, T, A, G, C, right? So it's just based on that complementary base pairing of A's with T's and C's with G's. So what's going to happen is replication is going to keep proceeding along this replication fork. It's going to continue to get opened up or unzipped, and the enzymes are going to continue to come in and add new bases based on the complementary base pairing until eventually the entire chromosome has been replicated. And then you end up with 2 double-stranded DNA molecules, 2 chromosomes. So there's some rules that go with DNA replication and some enzymes that are involved that you need to know. We already saw mention of a few of them, like topoisomerase, gyrase, helicase. The other main player is DNA polymerase. This is the one that actually brings in the new nucleotides to make that new strand of DNA. But DNA polymerase can only work in one direction, which is the 5' to 3' direction. It's initiated by an RNA primer because DNA polymerase can't just add a nucleotide to nothing. It needs to attach the nucleotide to something. And so we'll see this little RNA primer acts as that something to prime it, right, to get it started. Because DNA is anti-parallel and DNA polymerase can only go in one direction, you end up with the DNA being replicated in opposite directions along those 2 anti-parallel strands. One strand gets synthesized continuously towards the replication fork. That's called the leading strand. The other strand is getting replicated from outside, I don't know what you want to call it, the opposite direction, I guess, of the replication fork. And that's called the lagging strand. And that one doesn't get synthesized continuously. It gets synthesized discontinuously. And it creates these little fragments called Okazaki fragments. But no worries, because there's other enzymes that come in. DNA polymerase, there's actually multiple of them. DNA ligase, these other enzymes come in, they take the RNA primers out, they fill in the little gaps, they join together all the Okazaki fragments, and you end up with a complete double-stranded DNA molecule that was synthesized what we call semi-conservatively, meaning that the one strand is retained or conserved and one strand is newly synthesized. And again, we'll look at this on a diagram in a minute and explain some of those things we just talked about. But before we get there, here's a table of the enzymes. So usually on the exam, I have maybe a matching question or something where you kind of match up the enzymes to their functions. So you don't need to know every single one of these on the table, but you do need to know the ones that are on the diagrams on some of the slides we already looked at and on the slide coming up for DNA replication. So definitely you need to know DNA gyrase, you need to know DNA ligase, DNA polymerases. We can skip the endo-exonucleases. But you need to know helicase. We'll skip methylase, photolyase. You need to know primase. We'll skip ribozyme. You do need to know RNA polymerase, actually not for DNA replication, but for RNA transcription. So we'll get there in a little bit. And then topoisomerase or gyrase. I think that's it. Those are the ones that you'll need to know. All right, so here's the diagram of DNA replication that can go along with, where'd that slide go? This one, with this info here. Okay, so you should be able to read this info and look at the diagram and kind of make sense of it. Okay, so let's do that. So here's where your DNA is still compacted, supercoiled, wound up, right? Here's where it's starting to get relaxed because of the topoisomerase and gyrase, and then the helicase specifically is actually, breaking the hydrogen bonds between the A's and T's to create that replication fork. So you have the replication fork here. This is DNA polymerase. So DNA polymerase will synthesize DNA in the 5' to 3' direction, which means it starts on the 3' end of the parental strand, and it proceeds in the 5' to 3' direction of the new strand being synthesized. Okay, so it's the light purple here. So that's called the leading strand because it gets synthesized continuously towards the replication fork. Because once it reaches here, these enzymes are going to do their job to keep moving along and opening up the DNA. Then as this DNA continues to get opened up, DNA polymerase gets to continue to move along and synthesize the DNA continuously. What they're not showing on this leading strand is there would be one little RNA primer over here that helped get the job started. They're not showing that, but they do show you the RNA primer down here on the lagging strand. Because the lagging strand has a lot of RNA primers. Every Okazaki fragment has its own RNA primer that had to be added to get the Okazaki fragment started. So this is called the lagging strand, and it's synthesized discontinuously because it has to work from inside the replication fork. So this segment of DNA got opened, so the primase enzyme jumps in, adds an RNA primer, then DNA polymerase jumps in, starts adding nucleotides. But then it runs out of DNA that's opened for it to work. It would reach the endpoint over here, right? Because actually this was the first one that was synthesized. So then as more DNA gets opened over here at the replication fork, it has to start again. So another RNA primer is added. Another, DNA polymerase comes in, synthesizes another one of those Okazaki fragments. Bam, it runs out of, empty space to add more nucleotides. So again, it has to then jump back to where the replication fork has now further moved and opened up more DNA. And same thing, new RNA primer, DNA polymerase starts synthesizing again until it runs out. And it keeps having to do that and jumping back as more DNA gets opened up at that replication fork. So that's what we mean by synthesize discontinuously, creating Okazaki fragments. And the whole reason for this is because DNA polymerase can only synthesize in that 5' to 3' direction. And because DNA is anti-parallel, that's just how it has to happen. But again, No worries, because in the end, you've got DNA polymerase, DNA ligase that come back and fix and fill in all those little gaps and remove out those RNA primers. So DNA replication is also called DNA synthesis, right? It's something is being made. What happens anytime you make something? Well, it takes energy, right? So DNA replication requires energy. Where does the energy come from? ATP. Okay, so that's what it's showing here. We really don't need to get into all the details of it, but here's your ATP, or it's a nucleoside triphosphate, right? That's really the meat of ATP is really those 3 phosphate groups. That's what gives the energy. So this triphosphate comes in, there's some, bond breakage here, and one of the phosphates is broken off, and that's what actually powers the synthesis. That's what actually gives the energy. We'll talk more about ATP in chapter 5. So remember, we've been looking at diagrams that have showed it in a linear fashion, right? We were looking at just a little piece of DNA. But remember, this is happening on the entire bacterial chromosome, which is circular. So here's a view to see how that works. So on that circular chromosome, there is an area where replication starts. It's called the origin of replication. You actually get 2 replication forks that form as it moves around the circle. So eventually, when DNA replication is complete, you get these 2 double-stranded circular pieces of DNA. And I wish they did the colors more clearly, but the parental strand is dark purple, and the new synthesized, what we call daughter strand, is light purple. And that shows that semi-conservative nature. One strand is conserved, one strand is newly synthesized. There is proofreading mechanisms built into DNA polymerase, so this process is highly accurate. It's not that common to get mutations, to get mistakes during DNA replication, because when they do happen, they can get repaired.
In order to pass genetic information on to its offspring, an organism must make a copy of its DNA. The process of copying DNA is called DNA replication. During replication, each strand of the parental DNA serves as a template in the creation of new DNA. Because each newly synthesized DNA molecule is made up of one parental strand and one new strand, DNA replication is described as semi-conservative. One strand in each molecule is conserved, while the other strand is newly replicated.
Okay, that was short and sweet, but here's one that's going to be a little more detailed.
Enzymes called DNA helicases use energy from ATP to break the hydrogen bonds that hold the 2 strands of the DNA molecule together, causing the DNA to unzip. The site where DNA helicase unzips the DNA is called the replication fork. As replication continues, these enzymes continue to break hydrogen bonds, and the replication fork moves ahead of the newly synthesized DNA. Stabilizing proteins bind to the single-stranded DNA, holding the strands apart and protecting them from degradation.
Actually, that wasn't the one I thought it was going to be. I think it might be this one. It's a little more detailed.
DNA polymerase can only add bases to the 3' end of the nucleic acid, which means DNA polymerase can only add bases to a nucleic acid. It cannot start replication from scratch. Cells use an enzyme called primase to synthesize a short piece of RNA called an RNA primer. This primer provides a 3' end on which DNA polymerase can act. An enzyme called DNA polymerase catalyzes the addition of bases to the 3' end of the growing DNA molecule. This reaction is powered by the energy in the high-energy bonds of the nucleotide triphosphates. Hydrogen bonding between the parental strand and the nucleotide triphosphates dictates which base is added. Only thymine fits correctly across from adenine, and only cytosine fits correctly across from guanine. An enzyme called RNase removes the RNA primers, leaving a gap in the DNA which can be filled by DNA polymerase. DNA ligase seals the maps between the DNA fragments created during synthesis of the lagging strand.
All right, so, So that was DNA replication. Hopefully, it wasn't too painful. We did hit 30 minutes right now in the recording, so I'm going to stop this one, and then we will have a part 2 where we will pick up with transcription.